Bacterial cells have long been considered simple organisms, but recent research reveals a surprising complexity. A new study from Northwestern University and the University of Texas-Southwestern has discovered that bacterial cells can remember temporary changes in their environment and pass these memories to their offspring. This memory transmission occurs without altering the genetic code, challenging our understanding of heredity. The study, published in Science Advances, highlights how bacteria can inherit traits through mechanisms beyond DNA, offering new insights into cellular behavior and potential applications in medicine. Traditionally, it has been assumed that heritable traits in bacteria are determined primarily by DNA. However, this study challenges that notion. A central assumption in bacterial biology is that heritable physical characteristics are determined primarily by DNA, said Adilson Motter, the study's senior author. The research suggests that information can also be stored in the network of regulatory relationships among genes. This discovery opens up the possibility that traits can be transmitted from parents to offspring through changes in gene regulation, rather than genetic alterations, providing a new perspective on inheritance. The study delves into the concept of regulatory networks, which function like communication systems among genes. These networks allow genes to influence each other, creating a complex web of interactions. The research team hypothesized that these networks could hold the key to transmitting traits to offspring without genetic changes. We found that temporary changes to gene regulation imprint lasting changes within the network that are passed on to the offspring, explained Motter. This finding suggests that the echoes of changes affecting parent cells persist in the regulatory network, even as the DNA remains unchanged. To explore their hypothesis, the researchers turned to Escherichia coli, or E. coli, a well-studied model organism. E. coli is a single-celled bacterium with approximately 4,000 genes, making it simpler to study than more complex organisms. The team used a mathematical model to simulate the temporary deactivation and reactivation of individual genes in E. coli. These simulations revealed that transient perturbations could lead to lasting changes, which are projected to be inherited for multiple generations. The researchers are now working to validate these findings through laboratory experiments using a variation of CRISPR technology. The study proposes that reversible stress can trigger irreversible changes within the regulatory network. When a gene is temporarily deactivated, it affects neighboring genes, creating a cascade effect. By the time the initial gene is reactivated, the network has already undergone a self-sustaining transformation. It's a network phenomenon, said Motter. Genes interact with each other. If you perturb one gene, it affects others. This chain reaction suggests that non-genetic heritability could be a common phenomenon, as similar regulatory networks exist in other organisms, potentially leading to widespread implications across biology. The discovery of non-genetic heritability in bacteria has significant implications for medicine and beyond. By understanding how bacteria transmit memories, researchers could develop new strategies to combat antibiotic resistance. For instance, subtly altering a pathogenic bacterium's regulatory network could render its offspring more sensitive to treatment. Additionally, this research could inform studies on non-genetic inheritance in more complex organisms, providing insights into human health and disease. As Motter noted, in biology it's dangerous to assume anything is universal, but this study opens the door to exploring new avenues in genetics and cellular biology.